Let us discuss Biology Paper 2 KCSE prediction. On to question 1. A student investigating an aspect of photosynthesis set up an experiment as shown in the diagram below. Okay, so we have a bell jar. Within it, we have two things. We have a potted plant, a plant in a pot, and the pot is covered by a polythene bag. We also have a burning candle. Interesting. The bell jar was made airtight, so that means that there could be no gases leaving or entering. After some time, the candle went off, as expected. The student placed the setup in direct sunlight for 5 hours. Part A. Give a reason a burning candle was included in the setup. Let's look at this. When we talk about a candle is a hydrocarbon. It consists of carbon and hydrogen only. So when hydrocarbons burn, what happens is that, of course, they're going to use up oxygen, but they will also release carbon dioxide. So in this case, the reason why we are including a burning candle is so that it can supply carbon dioxide. Remember, the question is saying that we are investigating an aspect of photosynthesis. This, of course, relates to the carbon dioxide. B Roman 1. What will be the difference in the composition of gases in the bell jar at the time the setup was placed in the light and after 5 hours? So the question is asking us for the composition of the gases before and after. Now, which gases are we referring to? There are actually three. Oxygen and carbon dioxide are the obvious ones, but we also have water vapor. Remember, water vapor is a component of air. Now, at the beginning, we're going to have more oxygen, very little carbon dioxide, and also less water vapor. Now, after 5 hours, what we would expect is that the carbon dioxide will be even more reduced, oxygen would have increased, and there will also be more water vapor. Now, let's look at Roman 2, where I'll be explaining why we have these changes. Give reasons for your answer in B1 above. Now, if you look at our setup, we are going to have three different processes taking place. Number one is you're going to have transpiration. Now, transpiration is the process whereby plants lose water to the atmosphere in form of vapor. So water vapor is going to be present before, but when the plant carries out transpiration, you're going to have more water vapor being released, and therefore this will cause an increase. Moving on to the next process, this has to do with combustion of the candle. Now, as stated before, the candle is a hydrocarbon. On burning, it's going to lead to the production of carbon dioxide. So, as expected, you're going to have oxygen being used up and carbon dioxide being produced. But this carbon dioxide will then be used by the plant to carry out photosynthesis. So you're going to have the plant carrying out photosynthesis, utilizing the carbon dioxide within and producing oxygen. So that is the reason why at the end, you're going to have more oxygen, but even less carbon dioxide. Part C suggests two reasons why it was necessary to cover the pot with a polythene bag. Okay, now reason number one has to do with the microorganisms that are present in the soil. Now you're going to have microorganisms naturally present in the soil, and these are organisms, so they're going to carry out respiration, and by doing so they're going to use up oxygen, release carbon dioxide. So in short, this will actually interfere with the composition of the gases. You need to know conclusively that whatever results you have are actually from the processes you intended, from the candle and the plant, and not from anything else. So you're going to cover it with a polythene bag, to prevent any interference with the experimental results. Number two is to prevent evaporation of the soil water into the bell jar. Question 2. The diagram below shows a transverse section through the proximal convoluted tubule of a mammalian nephron. Part A, name the structures labeled A and B. Okay, now let's look at the diagram. Let's look at this part. So that is actually a part of the nephron. So the nephron is the basic functional unit of the kidney. When we talk about the kidney undertaking different processes such as excretion, these are usually carried out within the nephron. Now, the nephron has many parts. The part we are focused on right now is the PCT, the proximal convoluted tubule. So, we are zooming in, and when we do so, this is the image that we are going to obtain. Now, if you look at our image, we have this part which is labeled as 2, Roman 2. If you look at it closely, you're going to note that this is actually a part of a blood vessel. Why am I saying this? It's because of this cell. This cell looks so close to what a red blood cell should look like. It's biconcave in shape. It's almost red in color. So this is a red blood cell. And if this is a red blood cell, then that means this is a blood vessel. By the way, if you look at the nephron, the part that we have over here, you're going to note that it's highly vascularized. It's surrounded by a lot of blood capillaries. So Roman 2 is a blood vessel. Now, over here, these are epithelial cells. They are found in the PCT, the proximal convoluted tubule. Now, these finger-like projections that are found at the end are microvilli. Now, whenever you hear or whenever you see that a certain part of the body has microvilli, then you should know that the reason why they are there is to increase the surface area for whatever process is taking place there. Now, in this case, which process occurs in the PCT? We have selective reabsorption. This is the process whereby useful substances such as glucose, amino acids, and such are reabsorbed back into the body. Okay? Now, this happens through the process of active transport. Now, when it comes to active transport, you're going to have substances moving against the concentration gradient from a region where they are at a low concentration to a region where they are at a high concentration. And this process requires energy. This energy is usually in the form of ATP, adenosine triphosphate. And that is the reason why you are seeing this. You see this ATP? Over here we have ADP. This is yet to be converted to ATP. So it's actually a hint that informs you that whatever is happening here requires energy. And since we only have 3 physiological processes, diffusion, osmosis, and active transport, and only one out of these 3 requires energy, this process, of course, is going to be facilitated by none other than active transport. Now, this is also the reason why you're going to have numerous mitochondria. These are the organelles labeled as B. So they are numerous in order to provide enough energy for active transport to take place. Part B, set 3 ways in which the tubule is adapted to its function. Okay, so how is the PCT adapted to its function? What did we say its function was? Selective reabsorption of useful materials. Now, I've already mentioned 2. You're going to have numerous microvilli to increase the surface area for reabsorption of materials. Number 2, you're going to have numerous mitochondria to supply energy for reabsorption of useful materials, of course. And lastly, if you look at the PCT, you're going to note that it's coiled. Now, the reason why it's coiled is so as to slow down the movement of materials, okay? So the materials are still going to move, but at a slower rate, taratibu. Now, by doing that, you're going to have more time allocated for the reabsorption of materials. Now, I want to say this. When it comes to KLB, in some instances, you're going to note that these 2 characteristics, length and coiling, are usually associated with one another. They're just lumped together. For example, you're going to be told that the PCT is long and coiled in order to provide a large surface area for reabsorption of materials. This is wrong. Those are 2 different characteristics. When it comes to it being long, yes, that is for increasing the surface area. But when it comes to coiling, that is to slow down the movement of the filtrate so that you can get, more time for reabsorption. So 2 different properties. Part C, name 2 substances that move from region 1 to region 2 during selective reabsorption. Okay, so we are talking about substances that have been reabsorbed from the PCT to the blood vessels so that they can then be transported back to the body. Glucose, amino acids, fatty acids, and glycerol, Any of those is correct. Roman 2, the physiological process in the activity in C1 above. So the physiological process involved in reabsorption of materials in the proximal convoluted tubule is active transport. Question number 3. Inheritance of human blood groups deviates from normal monohybrid inheritance in 2 ways. State the 2 deviations observed. Okay, now let me explain. So when it comes to monohybrid inheritance, this is whereby you're going to have a single characteristic controlled by a pair of alleles, by 2 alleles. Now I'm going to give a very common example, and that is with regards to the pea plants. So when you talk about height in pea plants, tallness, dwarfness, we talk about 2 alleles that control height. So it's either TT, caps, or TT small, or it can be heterozygous, where you're going to have 2 different alleles as such. But in all 3 cases, you're having a trait, which in this case is height, being controlled by 2 alleles. This is known as monohybrid inheritance. Now, when it comes to the human blood group system, it's different because number 1, it's controlled by 3 alleles. So you're going to have 3 different alleles. You're going to have one allele that is responsible for the presence of antigen A. So for example, people who have blood group A are going to have the allele that is responsible, for controlling antigen A, for the presence of antigen A. And then you have another allele that determines the antigen B. And then you're going to have a third allele that is responsible for no antigens. So such people are of blood group O. Now, their alleles lead to the production of no antigens on the red blood cells. Now, a person can either be blood group A, so they're going to have the A alleles, or they can be blood group B, or can be blood group O, or the last one, whereby they can be AB. So they can have 2 alleles. So one allele is responsible for antigen A, and another allele is responsible for antigen B. Now, one interesting thing about these 2 alleles is that they are codominant to one another. So that means if they are present, they will both be expressed equally in the phenotype. So if a person has the genotype AB, they are also going to have blood group AB. So they're going to have antigen A as well as antigen B in their red blood cells. So they are codominant. That of O is recessive to both A and B. Part B, what is sex linkage? This is the occurrence of genes on the sex chromosomes. Now, when it comes to sex chromosomes, we have the X chromosome and the Y chromosome. So, females have 2 X chromosomes. Males, on the other hand, have a single X chromosome and 1 Y chromosome as such. Now, when you have genes located on these chromosomes, they are then going to be transmitted together with the sex chromosome. So, they're going to be transmitted with the chromosomes that determine the sex of an individual. Part C. In Drosophila melanogaster, these are the fruit flies, the inheritance of eye color is sex-linked. Okay, let's stop there. So this means that the alleles that are responsible for determining the eye color of the Drosophila melanogaster are present on the sex chromosomes. Are they going to be present on the X chromosome or the Y chromosome? X chromosome most definitely. Whenever you see a question such as this, and it's talking about eye color in fruit flies, then this allele is present on the X chromosome and absent on the Y chromosome. The gene for red is dominant. Okay, so if the gene for red eye color is dominant, that means that of white color is going to be recessive. A cross was made between a homozygous red-eyed female and a white-eyed female. Work out the phenotypic ratio of the F1 generation. Use capital R to represent the dominant gene. Okay, now, first things first, we have to identify the phenotypes. Okay, actually, we don't need to identify because we've already been provided with that. We need to determine the genotypes. That's what I was saying. Okay, so when it comes to the female, that is our female, and it is a red-eyed female. The male is white-eyed. Now, moving on to the genotypes, we need to remember what we were told, that Actually, we need to remember two very important things. This is sex linked, so we have to include the sex chromosomes. So in case of the female, to indicate XX, in case of the male, XY. Now the next thing that we have to remember is which gene is dominant to the other. So if we look at the information, the one for red is dominant, the one for white is recessive. So whenever we have that of white, we are going to assign it a small r, okay? Now, the female is homozygous and red-eyed. So when it comes to homozygous, that means it has two identical alleles. And since it's red-eyed, that means both alleles will be responsible for the red eye color and therefore capital R as such. When it comes to the male is white. So that means it has this allele, the recessive allele. And since this allele is only present on the X chromosome, we are going to place it there. The Y chromosome will not have anything on it, okay? It doesn't have this particular allele. Now, once we have done that, we're going to separate them into gametes and then cross them. Now, on crossing, this is what we get. These are the genotypes of the F1 generation. So if you look at our females, they have the same genotypes. The males also have the same genotype. Now, let's start with the females. As stated, they have the same genotype and they are heterozygous. So they have one allele determining red eye color, the other determining white. But the one for white is recessive. So it's going to be masked. It's not going to be expressed in the phenotype. We will only see these females as having red eye color. The allele is going to be present in the genotype, but it's not going to be expressed visibly, in the phenotype. Now, what about the males? Let's see. These are also red-eyed. So actually, all the offsprings are going to be red-eyed. Sawa po? Yeah. So we're going to have two red-eyed females and two red-eyed males. Question 4a. Name the organelles that perform each of the following functions in a cell. Synthesis of proteins. These are the ribosomes. Roman number 2, transport cell secretions, Golgi bodies. Number 3, destroy old and worn out organelles or even the entire cell. That is the lysosomes. And the reason why they are capable of doing this is because they contain lytic enzymes. So the lytic enzymes can digest and break down organelles or even, the entire cell. Roman 4, package and transport glycoproteins. Again, Golgi bodies. Part B. Using a light microscope, a student counted 55 cells across a field of view whose diameter was 6,000 micrometers. Calculate the average length of the cells and show your working. Okay, now let me represent this information using a diagram. So we have that circle. I want you to imagine that circle as the field of view. Now, whenever you look through the eyepiece, you see this circle, that is your field of view. So we're being told that when a student looked through the eyepiece, they were able to see 55 cells across the diameter. From that point up to that point, there were 55 cells. Now, the actual diameter of the field of view is 6,000 micrometers. So if we want to calculate the average length of one cell, what are we going to do? We are simply going to take the diameter divide by the actual number of cells, and this gives us 109 micrometers. Part C. Why is it recommended to keep the stage of the microscope dry? Okay, so let me mention two reasons. Number one, to avoid refraction or reflection of light. Now, when this happens in the presence of water, what happens is that reflections or even refractions can occur, and they're going to make it difficult to focus and observe the specimen clearly. So this is the reason why the stage needs to be dry. Now, the second reason is you don't want to wet the slide. Why? Because if you do so, it can cause certain parts of the microscope to become wet, to be exposed to moisture, which can ultimately lead to corrosion. Question number 5. The diagram below represents a certain plant structure. Okay. Name the part labeled F. That is the stigma. B. Describe what happens when the pollen tube enters the structure labeled G. So what is G? That is the embryo sac. Okay. So when the pollen tube enters the embryo sac, what will happen is that the tip of the pollen grain is going to burst open. Okay. And the tube nucleus is going to disintegrate, to break down. This will allow the entry of the male nuclei. So how many male nuclei do we have? We have 2, hence the term nuclei. So a single is nucleus, more than one becomes nuclei. So in this case, we have 2. So one is going to fuse with the egg cell nucleus to form a diploid zygote. The other male nucleus is going to fuse with the polar nuclei to form a triploid zygote. Okay. Let me break it down for us, okay? Now, whatever is happening here is a process that is known as double fertilization. This is a process that is unique to flowering plants. Flowering plants are the angiosperms. When it comes to plants, plants have 3 divisions. Now, in the case of plants, a division is equal to phyla in animals, okay? So, the kingdom Plantae has 3 divisions as such. Now, when it comes to spermatophyta, the seed-bearing plants, it is further divided into 2 subdivisions. You have gymnospermaphyta and angiospermaphyta. Now, angiospermaphyta is what I'm talking about. These are the flowering plants. Now, double fertilization occurs in angiosperms. So in this case, if you look at the embryo sac, you're going to note that we have a total of 7 cells, okay? So we have 3 antipodal cells, we have 2 cyanogens, we have 1 egg cell, and lastly, we have a central cell, okay? So those are 7. Okay, let me go through them. So number 1, the cyanogens, there we have 2. These guide the entry of the pollen tube. That is their function. We also have 3 antipodal cells. These provide nutrients to the egg cell. And then we have the egg cell itself. Okay, this is like the VVIP, mwenyewe hapa, that, the one that is represented as J. And we also have another cell that is at the center. Within this cell is where you're going to have 2 polar nuclei. Now, out of these 7, the ones that we want to focus on mostly is the single egg cell and the 2 polar nuclei. So what happens is that from the pollen grains, you're going to have 2 male nuclei. Sawa? So female tunanini, we have 3 components. We have 1 egg cell and we also have 2 polar nuclei. On the male side, we only have 2 male nuclei. So one of the male nuclei is going to fuse with the egg cell. This will lead to the formation of a diploid zygote. Why am I saying that this is diploid? Because it's going to have 2 sets of chromosomes. One from the male nucleus and the other from the egg cell itself. So this is diploid. Now this forms the zygote, which will in turn lead to the formation of the embryo, okay? Now, in the other case, we're still remaining with 1 male nucleus, okay? So this is going to fuse with the 2 polar nuclei, leading to the formation of a triploid. Why triploid? Because it has 3 sets of chromosomes. 1 from the male nucleus and 2 from each polar nuclei, okay? So 3, triploid. Now this is going to lead to the formation of the endosperm. Now in case you're wondering, hey, what is with this process? This is actually a process that ensures that the endosperm, the one that is used for the storage of food, will only develop if the egg has been fertilized. So it's actually, a quite effective process. Okay, part C. What do the structures labeled J and H develop into after fertilization? So J forms the embryo and H, that is the integuments. So those become the tester, the seed coat. Moving on to the next question. In an experiment, 3 healthy rabbits were fed on equal amounts of carbohydrates. After 1 hour, the glucose concentration in the blood was measured at 30 minutes intervals for 3 hours. The results were as shown in the table below. Okay, so we have rabbit P, Q, and R and the mean. So A Roman 1, calculate the mean concentration of glucose in milligram per millimeter of blood 60 and 180 minutes. Okay, so at 60 and 180 minutes. Record your answers in the table. Now let's look at the table. So over here, we have rabbit P. Now this is the initial. So what is the initial? It means that this is the concentration of glucose that was measured in this rabbit 1 hour after it had been fed carbohydrates. Okay, so actually initial for all three was taken 1 hour after they had eaten. So, and they had eaten the same amount of carbohydrate. So that is not a factor. They ate the same amount of food, the same type of food. But if you look at their initial values, they differ from one rabbit to the other. Okay, we'll come back to this. So we have the initial. Na inaenda inapimwa after every 30 minutes up to the 180th minute, after 3 hours. Now I want you to look at the last row, the mean. If you're stumped, you're like, mean, nini. They've already given you hints, by the way. Look at this, 1.55. How did they get this value? They took 1.6 plus 1.49 plus 1.57, summed them, divided by 3, and voila, you have the mean. So in the case of the 60th minute, we are simply going to do the same. We are going to take the 3 values for the 3 rabbits, sum them together, divide by 3, give us our mean, which is 1.38. 180th minute, same thing. This will give us a mean of 1.06 milligram per millimeter of blood. On the grid provided, plot a graph of the mean glucose concentration against time. Okay, so there we have it. So for P, Q, and R. If you would like to look at the graph further, kindly pause the video and do so. Part B. What was the mean glucose concentration in the blood at the 75th minute? Okay, so what we're going to do is we are going to go to our graph, locate where the 75th minute is, it's here, and then move upwards until we locate the curves of the 3 rabbits, and then read the values on the y-axis. So we are going to take these 3 values, divide by 3, because the question is asking us for the mean glucose concentration, and that will give us that answer. Part C. Why was it necessary to use 3 rabbits in the experiment? Now the reason is so that we can get more reliable results. Part D. Suggest a reason why the initial concentration of glucose in the 3 rabbits was not the same. Now, if we look at the initial concentration, now a reminder, what is the initial concentration of glucose? This is the concentration of glucose 1 hour after the rabbits had eaten. Now, if you look at these 3, they have different values. So the question is asking, why will they have 3 different values when they were fed the same type of food and the same amount? Now, it comes back to the rabbits themselves. So number 1, different rates of metabolism, okay? Now, when it comes to animals, they have different rates of metabolism. You're going to have some having the ability to break down carbohydrates faster, therefore absorbing glucose quicker. So maybe this could have been the factor that influenced, the differences that we are seeing from the table. Now, the second reason comes to the rabbits themselves. Are we talking about rabbits that are of the same size and weight, or was one larger than the other? Are we even talking of rabbits that are of the same sex? Were they all female? Were they all male? Or were some male and some female? All of these are factors that can actually affect the metabolic rates and therefore affect the rate at which they absorb glucose. Part E. Account for the difference in mean glucose concentration during the period. Okay. Now, if we look at the mean glucose concentration, this last value, we're going to note that there is a trend, and the trend is there's a decrease. So we are having a decrease in the glucose concentration as time moves on. Now the question wants to know why. Why is the glucose concentration decreasing? Now the reason is because number one, glucose is going to be utilized by cells such as the muscle cells in order to produce energy. Okay. Number two is that some of the excess glucose is going to be converted by the liver to glycogen for storage, and some is also going to be converted to fats for storage. And that is the reason why the concentration of glucose decreases. And this is good. You don't want to have a continuously high concentration of glucose in the blood, just enough to be utilized by the cells. F. Name two end products of digestion: amino acids and fatty acids and glycerol. We are going to exclude glucose because it's already been mentioned. G. What is the fate of excess glucose in plants? Now, when we talk about excess glucose in plants or even in animals, you're talking about the same thing. What will happen is that they will need to be stored. Now, in the case of plants, they store their excess glucose in form of starch. So number one, you're going to have the glucose being translocated to storage organs, being transported to storage organs. Now, once there, it's going to be converted to starch for storage. Essay questions. Now, for the essay questions, what I'm going to do is I'm simply going to show you the marking scheme. Now, with question 7, the process of inhalation, I have discussed this extensively in a previous video of mine. Check it under the playlist of gaseous exchange and you won't be disappointed. So this brings us to the end of this wonderful lesson. See you next time when we discuss KCSE Prediction Chemistry Paper 2.